So viruses in general are obligated parasites. They need the host machinery for them to propagate because due to their small sizes, they cannot pack all the machinery needed for life, which is propagating genetic materials. So most of the viruses get in cells through, I would say, two major ways.
It seems that in many different types of viruses, the more transmissible the virus is, the less likely it tends to be lethal. And the more lethal it is, the harder it is for it to become transmissible amongst many people.
The best virus would be a virus that cause no symptoms. So they'll survive within our host with no symptoms, not much immune response, and they'll live with us. In turn, everyone will be infected. So that in this way that the species as a whole can exist for the longest time.
Shifting the topic a little bit from viruses to vaccines.
These kind of vaccines are safer because there is no chance for an active infection to take place. But because of that, they generate less immune response.
In what detail are we able to go in and excise a certain part of the genome and put in a different part that we're interested in for example, vaccine development? Dr. Xiao, welcome to the podcast. It's been a few weeks in the making, ever since you and I met at the Science to Business Network event back in October 4th, I think it was.
Yeah, that's right.
How did you actually gain a love for research? Why did you get into the position in the first place? Because right now you're a postdoc at Xiao, right?
Yeah.
So can you kind of just walk me along the path that you took to get to your current position?
Yeah, for sure. I think I've always had I think I was always curious. You know, my dad's engineer, and I had constant exposure to different things in science, whether it being physics, biology, and I think ever since I was a kid, I had, I was interested in science, and when I got into university, and I majored in biochemistry, which was an easy decision for me. But I was struggling between, biology or physics, but science nonetheless. And then I was second or third year, I took my first job at Health Canada.
Mm-hmm.
And I was hired as a co-op student, as a scientific evaluator. So I was doing a lot of analysis of toxicology reports. So that's, while reading those toxicology reports, I learned about how people were testing these chemical compounds, and then I was like, okay, maybe that's something I wanted to do. And then my second co-op job was with Spartan Bioscience right here in Ottawa. I'm sure a lot of people heard about it. Yeah. So I was involved in the development of their genetic testing reagents. And I really enjoyed the stay there. And then finally, my third co-op was at Xiao, and I had great fun there. I was collaborating with people. I worked on some interesting projects, and I think what's the most engaging was that I was getting results, and it kind of propelled me to study, read, and experiment more and more. And I think, and the work that I did in the co-op, got into a manuscript that was published, during my master's. And I felt a sense of achievement. So that kept me going. So yeah, so I've been in research ever since. I think that was almost 11 years ago.
Wow, time flies.
Yeah.
Something you mentioned earlier, that you ended up deciding between biochemistry and physics. What made you decide taking the biochem route? Was it your job at Health Canada, or was that before?
Oh, no. I did have interest in both, but because I took biochemistry as my major, and I was kind of like exploring in the realm of physics, and then like as I learned more about physics, I needed more math, and I just kept on accumulating credits after credits, and in the end, I think I I had to make a decision because I was spending 5.5 years in undergrad. I needed to make a decision to where to go in the future. So I dropped physics because a lot of my credits were accumulated for biochemistry. So I think it's a time that made the decision.
And then your current role at Xiao involves you working with different viruses, understanding more about how vaccines work. And so, what is your core tasks, like day-to-day tasks, and how did you end up at Xiao?
I ended up at Chio. Like I said before, I was a co-op student there before, and which because of my experience there, which made me decide to go back there for my graduate studies. So that's why I went back there, and I've been there since 2013 September. And my current role is as a postdoc research fellow, but my PhD was actually in cancer, and then I made a transition to microbiology, which is what I'm doing right now. And I work on developing vaccines that could potentially be delivered orally, which is which holds a lot of benefits to different populations. For example, the low-income countries, and also it's very easy to transport because oral delivery requires the vaccine to be more stable. So would it be more accessible to people? I think, yeah.
So, okay, there's quite a bit to unpack there. Because I'm curious about how oral vaccines work and how you even go by developing them. But before I ask you more about that, let's kind of unpack a little bit of the basics. So, can you tell us a little bit more about how a virus works, how it can get into the host, humans, for example, and how does it evade detection by the immune system?
Yeah, for sure. We've heard a lot about viruses since COVID-19. I'm sure a lot of people must be tired of hearing viruses now. So viruses in general are obligated parasites. So meaning that they need the host machinery for them to propagate because due to their small sizes, they cannot pack all the machinery needed for life, which is propagating genetic materials. So most of the viruses get in cells through, I would say, two major ways. So the one major way is getting to the cell by those surface receptors on the cells. So in our cells, mammalian cells, in the plasma membrane, there are all kinds of receptors. Those receptors are there for sensing the environmental signals. And those environmental signals will be propagated intracellularly. So that's why they exist. And the viruses can have recognition to specific receptors. And so that allows them to bind to the cells. And when that happens, the cell will take the viruses in by a process called receptor-mediated endocytosis. And another way of getting to the cell can be that viruses can be engulfed by cells through a process called phagocytosis.
Mm-hmm.
And once the viruses are inside the cell, they can just pop out and get into the cytosol of the cells and then start the whole infection process.
Now, one thing I'd want to know, this might be a question that many have on their minds. Is a virus considered a live organism? Is it a living organism?
Well, I would say that, yeah, it is a common question that I've heard many times. I think I would consider them contextually. So if we're looking at virus as particles by themselves, standalone object or beings, I don't think they are considered as life because they don't, like I said, they don't have all the machinery necessary for propagation. Mm-hmm. So they cannot replicate just by themselves without a host. But when a virus enters a cell, all of a sudden they can hijack all the cellular machineries for them to propagate their genetic materials and reassemble, replicate. And some viruses even hide inside cells for a long time. So I think when in that state, I think a virus, some of which are called provirus, they can be recognized as life because they have everything needed for life, which is propagating themselves and their genetic materials.
I suppose that definition only applies when they get into host cells.
Yes.
Because beforehand, that's not the case.
I don't think so.
Okay.
Technically.
What are the key ingredients, let's say, that viruses are composed of?
Right. So viruses are really simple creatures. For example, take COVID-19 as an example. So they have, everybody heard about the spike protein, which is on the outside of the virus. They're called spike protein because they are protrusions on the outside. And those spike proteins are actually anchored into a membrane, which protects the virus from the outside. And inside, there are their genetic materials. And genetic materials, I'm talking about all viruses, can be different. It can be, they're all nucleic acids, but there can be double-stranded DNA, single-strand DNA. There are single-strand plus single-strand DNA or minus, so in different orientation or of DNA. And there are RNA viruses, single-stranded, double-stranded. So they're all viable genetic materials. So they're all packaged inside. So for COVID-19, they have double-stranded DNA inside. And these are not free-floating inside. They're also bound by other proteins that they anchor the genetic materials. So they are almost attached to the inside of the capsid, which is what protects the virus.
Okay, so that's the enclosure that kind of houses
enclosure. That's right. Yeah.
And then you mentioned there's positive and there's negative form of DNA within the virus. So can you elaborate a little bit more on each?
So a positive. So DNA comes in different orientations. They are marked as 5', 3'. So a positive orientation is usually denoted as 5 to 3 because that's how proteins are being translated, how we read DNA, basically. But there can be also in the reverse direction. So what it impacts is that the processes of transcription would be different, which is the process of turning DNA into RNA. Because for a protein to be made, we need to generate messenger RNAs, which are naturally read from 5 to 3 direction. So there is a very famous scheme. I think it's called the Baltimore scheme, which is basically describing viruses as in how their genetic materials are turned into 5 to 3 orientation mRNA. So if you have a negative strand, single strand DNA, it needs to be turned into the right orientation in messenger RNA for it to synthesize the right proteins.
Right, okay.
Yeah.
Could we talk about the different kind of viruses that are out there and how different types have evolved different traits in order to infect humans and evade the immune system and to replicate further inside host cells?
For virus, when they get into the cells, due to our immune systems, we always want to fight them off, right? Because our immune systems are naturally defending us from all the foreign pathogens. But viruses have unique ways for them to shut these processes down so that either by blinding our immune system or making the infected cell harder for them to see. So there are many processes. Would you like a few examples?
Yeah, sure.
Yeah.
Actually, how do they blind the immune system from recognizing that there's a foreign body inside of the host?
Right. So we have in our cells receptors that are called pattern recognition receptors. So these pattern recognition receptors are specifically made to recognize known structures that are found on the pathogens, like polysaccharides, LPS, or part of a protein from a pathogen. So one of the processes for the immune cells to see them is that the immune cell needs to see what's being presented outside of the cells. And when they see something that's different, that's not from ourselves, they'll see it, and they can attack it and then kill the target. So the whole process is it's called antigen presentation, and these are done by molecules called MHCs, the major histocompatibility complex. So when a virus get inside the cells, they have to make their own proteins, and some of their proteins will be processed and degraded inside the cell, and these degraded fragments of the proteins will then be loaded onto those MHC molecules, and then they will traffic to the outside of the cells and the plasma membrane, and then presented to the immune cells. And the immune cells, you can think of them as little soldiers surveying the area. Once they see something, they'll alert other cells at the same time as they'll engage and kill the infected cells.
And they're alerted to it as because it's not supposed to be there. And then there's surface presenting antigens that so other cells don't have those, right? Our cells in our body, they don't share the same types of surface receptors.
There are two classes of MHC. The class 1 is pretty much on all the somatic cells. So all the cells have them. Okay. So they constantly presented these. But what the difference is that the presentation of new things. Okay. So that's what's being recognized by the cell.
I see. Okay. And so upon presentation, they can kind of tag them for degradation or tag them that, there's a foreign body here, get rid of it, essentially.
Exactly. That's what our T cells do.
And when it comes to viral evolution, for example, COVID-19, they had multiple different variants in a relatively short span of time. Is that a normal evolutionary progression of viruses evolving at kind of a rate of several different variants within a year, or was that kind of unique to COVID itself?
No. I think evolution is a constant battle between pathogen and the host, because the host will always evolve like us. We always have new ways to attack them, and for survival, the pathogens will always have new ways to evade them. So there has been this constant battle for eons. So it's not surprising that viruses like COVID-19. SARS-CoV-2, COVID-19 is the symptoms they cause. They evolve, and it is a common feature for all viruses. And what's unique about SARS-CoV-2 is that we have taken many drastic measures, for example, in the means of immunization to help protect us from those viruses, and which that's what we call like a evolutionary kind of pressure on the viruses. So the more pressure you add on to the virus, they tend to evolve more, right? So that's why I think it is important to understand the process of evolution, so that we know that we're not over-immunizing ourselves, so that the viruses become super viruses. So because at the end of the day, it takes time to make new vaccines, and people will run out of vaccines. For example, in the low-income countries, but the viruses keep evolving, so they always find ways to avoid them. And then all of a sudden, when you are lacking this immunity, they attack you, they amplify, and you'll get sick. Just like the same case as what we call those super bacteria, right? Yeah. So uncontrolled antibiotic usage will create those superbugs that they are resistant to many, let's say, common antibiotics. So it'll be harder for us to control them.
So then, in that case, a perfect example is bacteria, how we combat them primarily by using antibiotics. What about antivirals? Have they not gotten to a point where we can combat the viruses as they are already in the body using antivirals compared to antibiotics, for example?
So antivirus, it's a very intriguing question and a very technical. So, for example, how do you shut down a virus, right? So once they establish infection, they use cellular machineries for them propagation, right? The only way for us to shut them down once an infection has been established is to inhibit those natural processes that we have inside the cells. So, for example, the virus, once they get into the cell, they have to, for example, some viruses get into the cell by receptor-mediated endocytosis. They'll end up in those structures called endosomes. What we can do is that actually, one interesting process in the endosome is that the endosomes have those pumps that pump the protons inside, so making the endosomes acidic, and that activates enzymes in the endosomes, casseptins. Like these enzymes help virus in the process of getting out of it. Some viruses, not all. Let's say that we have a drug. I think chloroquine was one of the drugs that can do that. If we inhibit this process, you also inhibit the process for other cells to take in other things, can be other metabolites, nutrients. So you will end up having a lot of side effects. So it is incredibly hard to have an antiviral that has no or low side effects. But one better way to combat viruses is before they enter the cells. So, or we can alleviate symptoms once the infection has happened. So during early phases of COVID-19, I'm sure you have heard about people getting convalescent plasma.
Yeah.
So convalescent plasma it is from people who had prior exposure to COVID, to SARS-CoV-2. So once these individuals seroconvert, meaning that after infection, they generate antibodies, we can actually take these antibodies out, isolate them, and then inject them back into someone with severe symptoms. So that antibodies will look for and inactivate viruses that are already inside ourselves. But this way that we can eliminate or reduce virus spread inside our body, so that we can somewhat control the symptoms, which can be life-saving sometimes.
Yeah.
So that's the current state. I think that's why vaccination is quite important.
Mm-hmm. That's sort of a form. So you're basically taking these antibodies from a person that has already been infected, that already developed the defense mechanisms against the virus, and then putting them into a different person. And essentially, it is a form of vaccination, is it not?
That is, yeah.
Yeah, okay. I guess earlier when I asked the question about antivirals, it didn't occur to me that the biggest difference is that viruses have to replicate by using the host machinery, whereas bacteria don't. They can undergo binary fission, right, in order to split apart and make new bacteria, and then so on and so forth. So you can stop these kind of processes from happening, and you won't be damaging any healthy cells. But I guess this approach is more similar to what chemotherapy is. I understand there are different ideas, but it's the same thing. Chemotherapy will attack healthy cells and the cancerous cells, much like antivirals will inhibit functions that allow viruses to get in and other cell components to also do their regular functions.
Yeah, in a way, yes.
Okay. Then I want to kind of understand a bit more about how the virus lethality versus its transmission works, because it seems that in many different types of viruses, the more transmissible the virus is, the less likely it tends to be lethal. And the more lethal it is, the harder it is for it to become transmissible amongst many people. So why is that? Why is there that kind of dichotomy between the two different points there?
So I think it's a natural evolution progress. I think a virus being lethal is also partly dependent on the host immune response. Some viruses can cause major symptoms because we have major immune response against them. But if you think about in a evolutionary or species point of view, a virus, we always tend to be, let's say, they'll always want to survive more. For them to survive more, they must need to spread more. In order to spread more, they must cause less symptoms. The best virus would be a virus that cause no symptoms. So they'll survive within our host with no symptoms, not much immune response, and they'll live with us. And in turn, everyone will be infected. Or so that in this way that the species as a whole can exist for the longest time.
The viral species, you mean?
Virus,
right?
Yes.
Yeah.
So I think in a evolutionary perspective, I think there are advantages. It's more advantageous for virus to cause less symptoms, which is the natural progress of of COVID-19. We see the beginning, the Delta wave, they're more lethal, and then when we got to Omicron, they become less symptomatic, but they start to spread also more.
Right.
Too.
Yeah. You hear more people getting it at that point.
That's right. Yeah.
Okay. Then when it comes to viruses that are a lot more lethal, for example, like Ebola.
Ebola, yeah.
Yeah, exactly. It's the first one that comes to mind. So why did it develop these different traits that just make it so much more lethal rather than slowly evolving in order to become less lethal in order to be able to propagate more and survive for longer?
I think that's a good question. I think what we need to look into more would be lethality in terms of hosts. That's why I was saying in the beginning that it's more host dependent, because it may be lethal in humans, but it may not be in other species, so that they can propagate better in other species. But in humans, they do get, we call those crossovers. They can establish infections, but humans may not be the best host for them.
Okay.
Although they can infect, they cause drastic symptoms, and the host die, and they die along with the host.
So is Ebola zoonotic? Did it come from animals?
I believe so. Actually, yeah, I need to check into that as well and see. I think one interesting thing to check into would be what other species Ebola also infect.
So interesting. Upon a quick Google check, it seems to show that the reservoir host, or I guess host zero of which animal it came from, remains unknown. But the evidence and the nature of similar viruses, research believes that virus is animal-borne and that bats are most likely reservoir. So I guess that it most likely may come from bats, but they're still not 100% sure. I don't know if this is fully up to date. But it is interesting because the viruses, whichever viruses that we end up making vaccines for, they tend to have limited amounts of genes. And so I had thought initially that the reason why, they tend to be more leaning towards transmissibility or leaning towards lethality is because they have a specific amount of genes that they can pack into the virus structure, and they'll give up one for another. Is that true? Can they have infinite amount of genes?
I would say no, because viruses are limited. Like they're small in size, right? So their genome is limited by their size.
Yeah.
So the last step, let's say one of the last steps of replication for a virus is self-assembly.
Okay.
So if there's so much genes and it just takes so much space, they cannot even self-assemble at that stage. So I would say that there is a limitation in what kind of genes they can have in them. That's why viruses tend to have the most important genes, and some viruses have incredibly like well-organized, let's say, temporally regulated transcription events happening. So this definitely is something that is it's evolved over eons, billions of years, and that made the virus so efficient because they only package what's needed for them, and they snatch what's not essential.
Okay.
Well, essential for their replication, but that they can borrow, let's say.
And they're constantly undergoing mutations during the replication process, and those mutations, I imagine, are influenced by host defenses.
Yes. So like I said, the selective pressure on the virus would tend to make them more prone to evolve because they would want to evade such processes.
Right.
But just the DNA replication or the replication of the genomic materials, it's not perfect. So there will always be mistake happening. It's just like us.
Yeah.
So some opportunistic or let's say beneficial mutations may happen in a place in that helps this virus to survive, and then these populations will get much bigger after the mutation. So we then will have a virus with a brand new mutation.
Interesting. Okay. And do we know what the earliest form of evidence of viruses existing is?
There has been. I don't think it's completely known, but we know one thing. I believe that one question has always been asked is that as we know that when life started to happen on Earth, it's first in the form of single cellular organisms.
Yeah.
So the question is like, which one come first? Bacterium, single cellular bacterium, or viruses? So I think the prevailing theory is that bacterium comes first.
Because they're incredible at just dividing on their own, not needing any host, right?
Yes.
Right.
And what we know first is that in the oceans in early time, there the predominant amount of viruses even existing right now in the ocean. We have we have incredible amount of phages. So these are the viruses that infect and kill bacteria. So if you are asking, because my my current supervisor had asked me before the same question. Is there evidence in fossil evidence of viruses, let's say?
Yeah.
Well, I mean, there hasn't been any, but there are things that close to it. So I think in the Cretaceous period, which is marked by a sedimentation of gray or white sediments and materials, and these are believed to be formed by algae.
Okay.
And it's also believed that these algae were constantly turned over, and their turnover was mostly mediated by viruses.
Interesting. Okay.
So that's one of the explanations for why during the Cretaceous period there's such massive dying of those algae, and they were believed to be killed by viruses. But no one has ever, let's say, looked into these fossil evidence and see virus inside, because for virus to fossilize, I don't think the nature of how the virus so the biology or the architecture of the virus allows fossilization of themselves. So unfortunately, we don't have any evidence.
Is it because they're too small to fossilize?
Yes, and they're
Fragile.
they're very fragile. They're most like a protein or lipid based
Right.
particles. It's really really hard to fossilize.
Because there are many different types of bacteria that are resistant to antibiotics, why don't we start to develop more bacteriophage treatments for known bacteria that's resistant to antibiotics, that don't really respond well, or that tend to have a very high turnover rate of becoming resistant to certain antibiotics?
There has been a lot of studies on that. I think as a matter of fact, one of the labs at Xiao is studying that. Well, what their project is looking for bacteriophages that are naturally occurring in the wastewaters and select them for their roles to specifically infect and kill bacteria. And it is proposed one of the proposed therapeutic strategies for managing bacteria. Yeah.
So then we have to, is it possible, instead of looking for pre-existent bacteriophages, is it possible to make our own?
You mean synthetically?
Yeah, synthetic
Um,
bacteriophages.
I think it's definitely possible, but it'll take time to construct a phage that is specific to a very typical type of bacterium. Actually, I haven't checked, but because it's not my area of expertise. But one thing we need to be careful is that we want high specificity because we do not want to damage our natural microbiome in our gut,
Okay.
because it is very important for regulation of many different metabolic processes, also dietary antigens. So it is. So let's say if we have a phage that will make a phage that lacks certain specificity and that ended up killing all of our microbiome, which would be very detrimental to us.
Right.
So I think that would be a very important aspect of such research. I believe one day, like when we know enough, we would be able to, because the phage genome isn't humongous, so we would be able to identify those either specific signals or receptors or means for them to identify the right bacteria to infect and kill.
Sorry, it is or it isn't humongous, the genome of a virus?
It is not.
It is not. Okay.
It is not.
Okay,
It's
so
small enough for us to understand exactly
Right.
which piece does what.
Mm-hmm.
But think about this way. So if it's antibiotic resistance, right? Usually it's mediated by bacterium synthesizing a type of protein to counter the antibiotic.
Okay.
Like in the action of ampicillin, for example. So bacteria can make something that's it's called a beta lactamase, which would degrade ampicillin. But these are secreted molecules, enzymes in the outside of the bacteria. They're not something like a recognizable feature on the outside of the bacterium. So it'll be incredibly hard to target these, because somehow the bacteriophage need to recognize these signatures. For example, a bacterium that secretes X amount of enzymes that counter the antibiotic resistance genes or antibiotic resistance drugs, right? Somehow they have to recognize that first in order to kill the right bacterium. Yeah. Otherwise, this would be detrimental, I feel.
Okay. And a lot of our mental well-being and digestive capabilities relies on our gut microbiome. So
That's
that,
right.
I imagine, can be very, very devastating to the body.
That's right.
Okay. Moving on to a bit more about how virus genomes are edited. So how detailed or in what detail are we able to go in and excise a certain part of the genome and put in a different part that we're interested in for example, vaccine development?
Oh, we can be very, very precise, because through the sequencing technology, we know exactly what these genes sequences are. We can edit a specific gene from the virus to make them less lethal. We can add in a specific gene, which is called a transgene, to make virus express it, which may lead to some beneficial effect when we are utilizing viruses for therapeutics, for example. So this can be very, very precise. What we can do right now is using molecular biology techniques to insert transgenes inside the viruses. And these transgenes can be genes that stimulate immune responses so that we can use those viruses to enhance our immune response to, let's say, cancers or a certain type of a disease, which we can do right now.
Okay. Because one of the thing I was wondering is there are viruses such as the herpes simplex virus or HIV, which are not immediately detectable. And what I mean, like they kind of remain dormant, perhaps for a certain amount of years or a certain amount of months. Whichever duration that they're dormant for, they tend to become more active when something activates them. So first off, how does that process work? How much do we know about activation or what really triggers those types of viruses to begin replication when they've been dormant for X amount of time?
Right. So some processes, we know some, but I don't think we completely understand. Let's say take HIV as an example. So when we get infected by HIV, which targets predominantly T cells and some monocytes like macrophages, when they infect the cells, they can have their genomic materials incorporated into our genome, which will stay silent for years, and that integrated genome, which is just basically double-stranded DNA sequences, that is referred to as a provirus. So the provirus can stay with the cells for indefinite times until a signal hits. So I think I'm sure there has been a lot of updates, but one of those signals is that we know that for HIV to reactivate, they tend to be reactivated when the cell they infected in also start to reactivate. For example, like those T cells, when the T cells become reactivated, some signaling, especially transcriptional factors, will also be reactivated, such as MFAT, NF-kappa B. These transfection factors, they'll actually at the same time reactivate HIV provirus by transcribing the genetic materials. So that's one way for HIV to reactivate. And I'm sure there are other signals the viruses can take or to sense, and then they become activated again. That's just one of many processes.
Something I wanted to clarify a little bit more. During the time when a virus is infecting or it's traveling to different cells in order to get in and infect them, at which point is it most recognizable? Is it before it gets into the cell, or is it after it gets in the cell? And does the cell itself on the inside of the cell, does it have any defense capabilities of recognizing pathogens or intruders? And can it mount any defense?
Well, the answer is yes and no. So if you're asking if detection of the virus before infection happens, this is when the virus is in our bloodstream, for example,
right?
The virus hasn't entered our cells yet. If there are any other ways to recognize and eradicate the virus, yes, we do have those ways to do that. And these are done by antibodies that bind to specific parts of the virus so that they'll render the virus unable to bind to their host cells. So that's one way. And then we have another system called complement system that can also either enhance the antibody effect, or then themselves are those what I called earlier the pattern recognition receptors. They can actually recognize the viruses such a way. And also on our cells also have those pattern recognition receptors on the outside that can recognize those pathogens. So that's part of our innate immune system. So that's the one that we're born with.
Okay.
And that tend to not change based on pathogens. They treat all the pathogens the same way, right? So they're not, the other one would be adaptive. So when the virus enters the cells, there are many other ways for the cells to recognize the virus again. So for a virus that enters the cells by receptor-mediated endocytosis, there are receptors, they're called toll-like receptors, that are inside the endosomes that can recognize part of the virus or the part of the virus genetic materials, and then start downstream antiviral immune response. Okay, so that's one way. That's still part of our innate immune system. Once the virus has established infection, their proteins, the viral proteins, will be made, and then through antigen presentation that we talked about earlier, then they can be recognized by our T cells, right? So this is called this now has become adaptive immune system has comes into play, right? So I would say that when a virus enters the cell, we have more means to recognize them than before they enter the cells.
When can we say that the virus has infected the cell? Is it when it gets into the nucleus and takes over machinery?
I would say infection happens when the virus has been taken into the cells.
Okay, so just as soon as it gets into the cell, that's technically the virus is infected.
That's the start of their infectious cycle.
Okay.
Technically, when a virus docks onto the cell surface, that's the first step.
Okay.
They can dock onto surface receptors on the cells, and the surface receptors may not even know that's a virus, because virus can have structures like those either on the spike protein or other parts of virus that mimic the molecule that naturally binds to the receptors. So for the receptors themselves, they don't know what's binding to them. But what they'll do is that they'll start to take the virus, whatever that's bound to them, in, because that's one of their natural function. That's how the virus got taken in. So in a way that, it's cells are kind of blind. They don't know virus are bound to them.
Right.
They're just got taken in.
And so then basically the cell has some arbitrary amount of time between the time when the virus docks, gets in, and makes the replication happens within the nucleus. Is that correct?
No, not all the time.
Not all the
The
time.
replication, which part of the replication? So if we're talking about virus assembly,
Yeah.
it usually happens in the cytosol, right? Because that's where most of the translation happens. But transcription can happen either in the cytosol or in the nucleus, because some viral genomes get into the nucleus, some don't. So they can happen in both places. But when they start to make viral proteins, it's in the cytosol, because that's where translation happens, and that's where also viruses would assemble. Sometimes the virus would assemble as a whole, completely inside the cytosol. Sometimes they'll have proteins that are made and direct these proteins to the surface of the host cell, and then other parts of the virus will be recruited to the cellular membrane that are now covered with part of the virus, and then the virus will bud out. This is a process called budding out.
Right.
And then the virus will bud out as part of the and use part of the cell, which is their plasma membrane, as part of its own structure.
Okay.
Yeah.
So then I didn't know that actually before, because as I understood it, for us, whenever we're transcribing any genes, transcription specifically happens within the nucleus. And then anything after that involving translation, the messenger RNA is now in the cytosol, and it gets translated, or any additional modifications happens within the cytosol. Is that correct?
Yeah, that's the current understanding.
For us, at least, for our genes. And then I thought that because of that, the virus has to also transcribe its own genome within the nucleus, and then anything else involving translation would happen outside. So that you're saying that there are viruses that could do either or. They can transcribe also in the cytosol instead of in the nucleus?
There are different. For most the DNA viruses, their transcription are happening in the nucleus.
Okay.
So, but if I remember correctly, there are a subset of DNA viruses that could also do it in the cytosol, given the if they carry the correct RNA, the DNA polymerase.
Mm-hmm.
So if they have, for example, some polymerase are transcribe DNAs in the cytosol because the polymerase exists in the cytosol.
I see.
But most of the RNA viruses, because their genome is already RNA, so they will transcribe and translate both in the cytosol. So not necessarily the way that our cells are doing.
I understand. Okay.
Yeah.
And then during this process where they overtake our cellular machinery, can our cell still have some form of access to that machinery in order to do regular cell functions to maintain the cell as it's being taken over by this unwanted guest?
Actually, one of the first things that the virus would do once they infect the cell is to shut down such processes, because they want to use the cellular machinery to do their own to make their own proteins. But the cells, so I would say that in a way that the cells are making less of their own proteins, but most of the resources are being hijacked to make the viral proteins and whatever the virus dictate the cells. But collectively, because this is happening collectively, because the cell are making more viruses, they tend to be more metabolically active, because all of a sudden the cells have uncontrolled translation happening, and they're using a lot of energy. So they are also requiring a lot of energy. So to fight back, cells will try to shut these processes off as a mechanism to defend themselves from the pathogens. So some cells, some pathways will, let's say, stop translation altogether, even for their own self, for self proteins, so that they stop translating viral proteins, and some cells will just die. They'll induce cell death, so that there will be one less host for the virus to infect, so that the viral progeny it will be less.
So upon cell death, does the cell burst?
Well, if the cell can actually die by many processes, but if they decide their own fate, they can die by a process called apoptosis. So that's a more silent way of dying. So the cells basically chew themselves up, right? But due to an infection, some other ways of cell death could happen. So there could be a necrotic type of cell death where cells just burst open, and it's incredibly inflammatory, and there are viruses themselves can actually burst the cell open themselves. So it's called the lytic pathway of the virus release. So as one of the defense mechanisms, the cells tend to either shut down themselves, like flipping the switch, that's it, or they die, they kill themselves.
Interesting. And then in that process, when the viruses are continuously replicating, how do they know that they reached a point where we have made enough progeny and now it's time to burst out and infect other cells?
Well, some viruses would actually sense the level of the viral protein. There are specific viral proteins that they can sense. Then they decide the fate of the virus. For example, there's a protein called TAT in the HIV virus. So when this protein gets accumulated to a certain amount, some viruses may decide to get incorporated into the genome and stay silent.
Interesting. Okay.
But for most of the viruses, I think it's that they'll just keep replicating till the cells cannot hold themselves anymore, and then they burst and die.
Okay. The viruses die, or the cell dies?
The cell dies.
The cell dies.
Yeah.
Okay. And then viruses can go on infecting others.
Exactly. Yeah.
Okay. Now shifting the topic a little bit from viruses to vaccines, I think vaccines are very important because they treat a lot of conditions that would otherwise be very fatal, like polio or smallpox. Many other ones that don't even come to mind right now. meningitis, tetanus. Anytime I get, because I've seen videos and I've seen images of people that suffer from tetanus, and anytime I get a puncture wound, I like outside, for example, of something rusty, I freak out because I got my tetanus shot in 2012, and so I only got it recently again this year. But there was a window of time where I was out of that 10-year period. So anytime I get a puncture wound, I would freak out that I'm going to get tetanus. And so I am thankful for vaccines in many ways. And I want to understand how they work better. So first of all, what were the initial vaccines used on humans, and what are the different types that there are, and how effective are these different vaccines at treating the condition?
Right. So the earliest vaccines would be viruses themselves. So upon infection, you would develop immunity towards this virus, so that if you survive it, right, if it doesn't cause detrimental damages to your body or life-threatening diseases, you survive it, then you're immune to this type of infection, right? So that's the natural response of our body to viruses or other pathogens.
So that was the first vaccine that was used, to just having live viral pathogens within a syringe, and they just inject the vaccine?
That's right. We the first amongst the first few vaccines would be attenuated viruses, which is the ones that cause less diseases.
Okay. The
So
weakened ones.
weakened viruses, so
Right.
we put them back into people, and
Okay.
then so that they can generate immune protection without causing diseases. And when an actual pathogen comes in, and then we'll be able to defend ourselves from them.
How do they make these viruses less capable of inflicting damage? How do they attenuate them?
So attenuation could be simply deletion of parts of the viral genes that are disease causing, or maybe some viral genes that make them less prone for replication or shutting down our immune defenses, so that they're more exposed. Because they're more exposed, we can detect and kill them easier, and we can actually elicit more immune response, so we have a better long-lasting immune protection. That's one way of doing this. So weaken the virus, we can delete parts of the genome, or we can modify parts of the virus. So we can modify the genome in terms that parts of the virus will also be modified, so that they will be less disease causing. So that's called attenuated, live attenuated viruses. But one easier way to do this is to inactivate viruses altogether. So that's also one of the ways that we used to for COVID-19 vaccines. So once a new pathogen is detected in the population that is causing a pandemic, we can isolate this pathogen, in this case the virus, and then we can grow them in the lab, and we can use them to we can actually use chemicals to inactivate them, so that they no longer become infectious, and then we can transfer these viruses back into people. And because they no longer infect, but they still retain the shape of the virus, like I said earlier, some viruses, because the shape is still retained, they can still be taken into the cells. And once inside cell, although they don't infect, they don't multiply, they can still be degraded by the cells, processed by the cells, and then their antigens, their proteins, can be presented by the cells. And in that way, they can generate help generate the immune response against them.
And so then what were some of the inactivation methods that they used in order to make these viruses inactive compared to the attenuated ones?
I think chemical inactivation would be currently, I think, the industry standard. Other inactivation methods that's commonly used can also be UV, because it disrupts the genome of the virus. So the virus they'll have a defective genome, so they won't be able to replicate themselves. So that's one downside of using the inactive virus vaccines, because not all the viruses are always, like, not 100% of viruses are always inactivated,
I see.
and there always a chance that one virus does not get inactivated, and then it all takes is just one virus to replicate, and then you can get sick. And the other thing is the complete removal of the chemical reagent that tend to, like, cross-link viral proteins. So that usually this process is also harmful for people. So although inactivated virus vaccine is the fastest way to make a vaccine, it's not always the best way to do it.
Okay. And could you use heat to inactivate these viruses?
Yes, you can, but remember, some of the viruses are bound by plasma membranes, which are very susceptible to heat. So when you boil the virus, the whole structure will get damaged. So what you end up is separated genomic materials and the parts of the viral proteins, which is in a way is the basis of a protein subunit vaccines, because you still have parts of the protein that may be intact. However, heat usually will also denature the proteins.
Okay.
So you may get much lessened immune response, because the protein, although they come from viruses, they no longer have their tertiary structures. So they're not the ideal ways to generate neutralizing antibodies.
Okay, and then the next the next iterations of vaccines involve, after the inactivated ones, involved removing parts of the genome that makes the virus make people sick.
That's so removing parts of the genome can actually generate the attenuated viruses.
Okay.
So these viruses, they're alive. They can be used as a vaccine. They infect people, and which means that you have stronger immune response, and because they are actual pathogens, so they may generate a longer lasting immune response. So I think in this way they're superior to inactive virus. But also be careful that if you are still infecting people with a live virus. So it may not be the best way in terms of acceptance.
Okay. You mean like from general public?
From
to be
general
infected,
public,
right? Just the idea behind it. And so then, what was the next progression of vaccine development?
Right. So the next one would be using, let's say, parts of the virus. So like I said, we can recreate parts of the virus either by these are either by, like, synthetic technologies that we make parts of the protein from the virus that are known to generate a strong immune response, and then we put that into people.
Okay.
Or we make a virus-like particles that they lack all the genomic materials. They're just a little empty shell, and then we administer that into people.
Okay.
So they're both protein-based vaccines. So these kind of vaccines are safer because there is no chance for an active infection to take place. But because of that, they generate less immune response, right? So usually the protein-based subunit vaccines, protein-based vaccines need something called an immune adjuvant. For example, an aluminum gel to absorb these proteins or viral proteins in, and then administer to the people. And the adjuvant, which in this case aluminum gel, would actually be the causing immune response. And together with the viral protein that's embedded, it generates immune system also towards that part. And then we have immune protection against a virus.
Why do you need an aluminum gel instead of just injecting the protein as it is into a patient, for example?
Because, like I said, a protein is not infectious. Such a protein subunit vaccine themselves are not infectious. They're more immune silent. So because they don't cause a a strong immune response, we tend to also generate less protection against it. So that's why we need help. We need something. One crude way is to use a bifurcated needle, which would be a regular needle with hooks. So when we actually jab people with this needle, it induces lacerations on our skin, which in turns cause inflammation, because skin is one of the most tissue that generates the most inflammatory response. Right. So in that way, we physically cause inflammation. But a less invasive way is to deliver it with a adjuvant.
Right. Okay.
That stimulate immune response at the same time we deliver the protein substitute vaccine.
And then those, I imagine, are not as good at retaining an immune response for a longer duration of time, because over time that kind of response is going to slowly wane, right?
That's right, yeah.
And so then what would be a better option in order to deliver a vaccine that will give more long-lasting effects?
We would still rely on active infections. Let's say, which is also why we need boosters, right?
Okay.
I think, I don't know if you are due for another hip boosters, because every 15, 17 years you may need one. So these are, these are, let's say, not long lasting in a way that our immune systems always tend to wane and over time. Let's say we generate a vaccine that's from let's say a virus that's not causing any diseases, and it establishes an active infection, and it lingers, but there's no disease, right? But at the same time produces the antigen that we need for a certain type of other pathogen that's actually disease causing. Then that would be kind of ideal, because that kind of infection would generate a long-lasting immune response, because the infection itself from the vaccine, which would be in this case live attenuated viruses, would be long-lasting.
Okay.
So that would be, to answer your question, that would be the way to do it.
And I imagine the probably the best one in terms of efficacy is to just be infected by the virus itself, and upon survival, would that be the best outcome for developing immunity against the virus?
Yes. So one thing with these active infections, they also, everything has some pros and cons. So when you deliver a vaccine using a live attenuated virus, you generate immune response against that virus as well.
Right.
Right. So the infection may be longer lasting. I mean, the immune response may be longer lasting, but after you are vaccinated, you generate antibody against them, against the virus, those are viral vectors, so that subsequent injections, if you need, for example, the adenovirus-based vaccines, AstraZeneca, they are when we get the boosters, the over time it will become less and less effective, because we generate immunity against the vector themselves. So, but we have other ways to overcome that. We can use a similar vector, but a different serotype, which is a type that generates a different type of humoral antibody-based immunity, so that the previous antibody immunity will not affect the second round. So right. That's one way of go about avoiding it. And because antibody-based response usually are not as long-lasting as T-cell-based response, so over a course of 6 months, 1 year, your antibody against the viral vector will also weaken. And maybe after that, your body will have less protection against the actual beneficial viral vector, so you can have another booster then. So that process can keep going.
You mentioned AstraZeneca's adenovirus. How does that work compared to a vaccine that targets T cells? Because you had mentioned that a response for T cells is much better and more long-lasting than something like antibodies.
Right. So a vaccine that we develop will uh, uh, always cause, I would say right now, uh, a B and T cell immune response. B cell immune response will help prevent the pathogen establishing infections. And these can happen like early on during early stages of transmission. But once a person is infected, then the intracellular pathogens are cleared by the T cells. So the our aim is to develop a vaccine that targets both immunity, not just one of them. If it's just a T cell based immunity, well, we'll always have infections happening. It's after infection that we start to kill the infected cells, so that we would have, let's say, people who have little to no symptoms but still carrying the virus because they're constantly being cleared, right? And then they are spreaders. They're silent spreaders. So they're more dangerous because we cannot identify them easily. But if we only target the B cell immune response, then we would lose it quickly. After 2-3 months, our antibody titer will drop down a level that is no longer effective. So that we're more susceptible to infection only then, and the protection only lasts 3 months, 3 to 6 months. That's not ideal, right? We want long-lasting protection, so we need both.
So comparing oral vaccines to vaccines that are injected, what are some of the benefits of one over the other, and why is it beneficial to try to develop something like a vaccine orally when you can just inject it, for example?
The vaccines that are delivered intramuscularly, which is usually the main mode of delivery right now, it induces very fast systemic immune response. That meaning like both T and the B cell mediate immune response. But we already have evidence showing that this type of immunity is more in a way restricted because we overlooked one important aspect of transmission, which is through our respiratory system, or some viruses can infect in our gastro-intestinal system as well. And in our respiratory and gastrointestinal systems, we have something that is called the mucus. And the mucus protects us naturally from infection. But we overlooked the mucosal immunity. So intramuscular delivery of vaccines often not lead to development of mucosal immunity, or they develop weaker mucosal immunity. So that is a problem for pathogens that transmitted using these systems. For example, for a flu or SARS-CoV-2, these viruses can survive in our respiratory system, but get attacked when they enter the bloodstream.
Okay.
So these are people who have no symptoms because they're protected, but still have live virus in their nostril or in your nostril cavity, right? Nasal cavity. So they can still spread these viruses just like normal. So that's what we want to target as well. So oral vaccines, they are excellent for inducing mucosal immunity, which will shut down the primary mode of viral infection, because that's the first stage of infection. For example, SARS-CoV-2, they enter usually through our eyes or places where there are ACE2 receptors, which people already know, and the nasal cavities. So they also infect our gut as well. So these are the places that we need mucosal immunity. We need to develop that as well. So having a virus that orally that infects our gastrointestinal system or our respiratory system will help us generate immunity in these systems, so that they can shut down the virus at the initial site of infection.
Is there a different approach to developing oral vaccines? Like, do you have to have them encapsulated in certain vesicles, or be able to deliver them to mucosal cavities in a way that's different than how vaccines, subdermal or intramuscular vaccines, are given?
Very different. The mucosal immunity is very different because we are constantly exposed to mucosal antigens like food or things that we breathe in, right? And most things do not elicit any type of immune response. Imagine that we eat an apple and then we have an allergic reaction. That would be terrible for us because we'll die,
Yeah.
right, from eating food. So our system have developed this immune response that are less likely to be generated by those pathogens, by those antigens, actually, by food or even live bacteria or viruses, because of the fact that it is just that much harder for them to penetrate the mucus. So if they cannot penetrate the mucus, they cannot establish an infection. They'll just get excreted out without doing anything in our body. So we're safe, that they cannot infect us. So that's why this is very hard to make a vaccine that can actually break, like you said earlier, this tolerance, right? Plus, the gastrointestinal system is very acidic, so a lot of things will not survive in these environment. But if you survive this environment, your reagent may not be able to enter the mucosa and then get taken by the immune cells residing there and then have an immune response. There are things that we can, yes, to answer your question, yes, we do need to encapsulate. If we take let's say, a non-infectious agent, they do need to be encapsulated before we get ingested, so that they survive the stomach acid. There are materials that are responsive for stomach acid. So depending on the acidity, the structures can, let's say you can imagine it as an enclosure. This enclosure can open or close. So you can have a controlled release of certain reagents. So you can survive, you can pass the acidic environment, but when you reach the places that the reagent is released, well, it has to pass the second layer.
Right.
So that is very, very hard to bypass. But what we are trying to do is to harness the power of a naturally occurring gastrointestinal virus to do this. And this virus, which is called Reovirus, REO, Reovirus. It's not a real or fake. So this Reovirus, so Reo is an acronym for respiratory enteric orphan virus.
Okay.
So this virus is capable of infecting through our respiratory or enteric system, which is our gut. And it is termed as an orphan virus because it doesn't cause any known diseases. Although high levels of infection may lead to symptoms like diarrhea, but it's not life-threatening or anything close to that. So we're harnessing the power of this virus as an agent because it has the capacity to establish infection in the gut, meaning that this virus can go through both the acidic environment and then establish an infection in the gut. So it is a perfect reagent for us to use to deliver the payload into the gastrointestinal system. Once the virus establishes an infection there, it can elicit immune response to the antigens that we put into this virus, which was molecularly engineered into the genome. So once the virus infects the gut cells, these antigens will start to be expressed, and then we'll have an immune response against these, and then in the end, we'll have immune protection against those.
So then, can these oral vaccines be used to treat something that's not in the gut? Could it be used to deliver it instead to somewhere in the lungs?
Yeah, you can use nasal sprays to deliver the viruses, but what we're testing right now is mainly through the gut, because that's where we want the primary protection is. But we also find out that by having the virus infecting the gut, we not only just generate mucosal immunity, we also generate systemic immunity as well. So we can detect neutralizing antibodies from the serum in the blood. So imagine that it's like one stone, two bird situation.
Right.
So once ingesting this vaccine, you have both immunity, which is our ultimate goal. So we don't need two different vaccines to do the job. One does two, and also this is a live attenuated virus, so it may cause also long-lasting immune response, and it does not need an adjuvant because it's a live virus.
Right.
It's self-adjuvanted, meaning that it generates inflammation by themselves. So and they don't cause disease. They're safe.
Okay.
And these viruses are very easy to transport. They don't need minus 80 freezers that the mRNA vaccines would need. So it is very ideal for the developing countries where they lack these transportation systems.
Thank you so much for your time. I really appreciate it. I really appreciate you coming out here today. And I wish you all the best in your career going forward. Hopefully we can have a chat in the next couple of years, and we would, get some updates from you, see how your research is going. And I wish you all the best. Thanks again for coming out.
Oh, thank you. It's been absolutely a pleasure talking with you, and I love talking about science. And I know that I will in the future, and I can't wait to have another conversation in the next few years and see where or how things goes and how the vaccine development goes. I'm sure there will be some exciting development in the future. And I wish you all the same, and best luck for your study and your career as well.
Thank you very much, and thanks for listening, everyone.